Okay, I hope you can see now. So we were discussing about uniparental stem cells, and we discussed about without fertilization, those that could give rise with fertilization. We discussed about PG, GG, AG, and GG and AG. There is some problem with the graphics tablet, but I hope you can adjust it. Okay, so we need to get used to some terminology as usual. That is, the gyno means female, or I might actually say as maternal, and andro is for male, or I could say it as paternal. And we have discussed about different ways of how uniparental embryos are generated. One of it is parthenogenetic, when you just take an embryo that is in meiosis 2 stage, and you activate it, and it has two sets of genomes, or how do you say it, have diploid state. Only thing one has to ensure that we have prevented the extrusion of the polar body, and then whatever develops, it's development of a diploid organism or an embryo from the same egg, from one single egg. It can be called as parthenogenetic. Gynogenetic is manipulation of further parthenogenetic embryo zygotes by exchanging the chromosomes as shown here. And in this case, in these sets, both the genome sets are coming from females only, or maternal genome sets only. Therefore, they are gynogenetic. In androgenetic, to develop that, we have to enucleate an egg and then perform double intracytoplasmic sperm injection. That means you deposit two sperms into the enucleated egg, and then they would develop into a zygote or embryoid, which would both are derived from paternal alleles, so we call it as androgenetic. Then we have discussed about how to develop it after with fertilization. Right, in the previous one, there is nowhere there is fertilization. Fertilization is the fusion of male and female gametes. So in this case, we could take fertilized embryos and then exchange the the pronuclei from between these zygotes to generate gynogenetic or androgenetic. In the next class, we will also learn about some of the molecular techniques, how to molecularly characterize these zygotes, whether they are gynogenetic or androgenetic ones. So I would ask you all to please review the study or understanding about PCR and also about primer designing. It would be helpful. Please make a note of it so that you will understand in the best possible extent in the next class. Okay, we also discussed if the uniparental offspring is identical to parent or not. It is not. It is highly similar, but it's not identical, because during gametogenesis there is chromosome exchange, or what do you say, the exchange of genetic material between non-sister chromatids, that is recombination during meiosis. Therefore, they are not identical. We also, I think, did we discuss this part or not yet? Can somebody tell me, please? Someone? Am I audible?
Yes, I didn't attend last class, so I'm not sure.
Okay, that's all
So
right then.
we have to start this. We didn't do this.
Say it again, please.
We didn't start this, sir.
Okay. So once we have made the zygotes, what can happen of them is the question. The developmental potential, what can they give rise to? So if it is a normal diploid zygote, it will have a certain amount of placenta and a certain amount of the embryo. But if you take the androgenetic one, the AG, sorry, the pen is having a little bit trouble. Let's see if it works. No, it is still the problem. Okay, the AG, I'll just use this. I'll just use the laser pointer. The androgenetic one, what happens is it has enormous placenta and the embryo is very small. Compare it with that of what would have been relatively of the normal embryo. At the same time, the gynogenetic one has a different problem. So here it is written gynogenetic and parthenogenetic. It is all right. They both are same, except that here in parthenogenetic one, both these sets have come from the same egg. Whereas in the gynogenetic one, both these sets, one set has come from one egg and a different one from the other. So they are different by the way we make them, but otherwise they are very similar. If you look at the developmental potential of them, in the gynogenetic ones, the placenta is undergrown, whereas the baby is relatively big, right? So there are problems. There is developmental defect in the way these zygoids, the uniparental zygoids, actually grow. Okay, that's the first thing. So we can never, to the current day understanding, we cannot get a complete viable or normal uniparental zygoids. Okay. So the next thing is we need to know what can be done with the embryonic stem cells that are derived from uniparental stem cells. So at this stage of blastula of these uniparental zygote or embryos, we could take out the ICM and make embryonic stem cell lines. But what to do, what can they give rise to is the question here. So first thing, the technique that is used is simple. It is to make chimeric mice. Chimera, you know that it is made up of two different organisms, one body made from two different organisms. It could be different species also, as where humans are obsessed all over the world. If you see in cultures everywhere, they have mythological figures in majority of the cultures, ancient cultures. So basically, it can also be in biology. When you say chimera, it means two different sets of or genome sets present in the same body. That means some cells have some types of genome, some genome set, and the other one has a different one. We will discuss this later too. So for now, what I would want you to understand is the technique. How can we make a chimera? We take a blastocyst, and we would have had embryonic stem cells. In this context, these embryonic stem cells are derived from the uniparental. These are uniparental embryonic stem cells. And then we inject them into embryos or the blastula at the blastula stage. And so some parts of the body is derived from the already existing ICM. Some parts of the body will be derived from ICM that we have injected, or the uniparental embryonic stem cells. So I hope you remember that ICM is pluripotent and it can give rise to every tissue in the body. All tissues that we have in our body are derived from the ICM. So once we have manipulated the embryos in such a way, these blastocysts are implanted into a surrogate mother. We would have done multiple of these blastocysts, and eventually the mother, the surrogate mother would give birth to litter or the babies, pups. And then these individuals would be chimeras, meaning that some parts of the body are made from the donor, this blastocyst ICM. Some parts of its body are made from the inserted uniparental embryonic stem cells. And therefore, this organism is chimeric. So the extent of chimerism can differ. That means in some of these pups could be having a large amount of red cells or uniparental stem cells, may have been derived from uniparental stem cells or the injected embryonic stem cells. And in some cases, the proportion could vary. Okay, so that is a chimera. And chimeric mice are very common in stem cell technology. They use it for many purposes. During this course, we will read a few more times about these construction of mice and so on. So how we can do and why are we actually doing these embryonic stem cells is there are multiple purposes. The most important one, of course, is the research part. And if we try to derive any embryonic stem cells, and probably we have already seen that AG and GG cells, androgenetic and gynogenetic ones, cannot give rise to a complete viable organism. But if you're looking at the embryonic stem cells, can they give rise to some tissues at least? That's the question, and that's where we are going through the chimera. So here are some of the results that people have found is that through using the chimera technology or so, they have found that androgenetic cells mostly give rise to mesodermal cell lineages. And they grow aggressively. They grow much more faster. And when you say they can readily transform, that means they become cancerous, right? They are not in the control of cell cycle regulation. Whereas GG or parthenogenetic ones or gynogenetic ones, they give rise to most ectodermal lineages like brain and so on, but very rarely of the mesodermal lineages. And they are very slow growing. Compare this. AG cells give mesodermal lineages, but not other types. PG cells give ectodermal or brain-like, but not mesodermal. And AG cells grow much more aggressively and fast, and they can become cancerous too. Whereas PG cells, they grow very slow. The problem is that because of genomic imprinting. In the previous course, we had a brief discussion about genomic imprinting. I hope you remember a little bit of it. We will try to discuss this now as well as many times during this course. So if you didn't understand this time right away, it's all right. We will try to eventually understand. That's what we are aiming for. So the concept of the focus of today's class is genomic imprinting. I would want to use my graphics tablet one. Well, so I have to use. So just give me a minute while I correct. While I try to fix my graphics tablet. Just give me a minute. Just wait, please. Just give me a minute. Okay, I hope it will work, or let me see. Sorry. Okay, hope it gets fixed. Let me try. No. Sorry. Okay, I will anyway repeat, so you don't worry about if we couldn't understand it all completely today. It is anyway not going to happen in one go. Usually, the concept for this will take a few times, so it's all right to take a few minutes. Okay. Maybe I have to reinstall this. I'll do that at a later time. Maybe I have to restart my computer, but I'll do that later. Let's just go ahead with this. Okay, so the point that I was discussing is about genomic imprinting. In the last class, if I have discussed, I will try again. There are two components about it. We have so many genes. Some genes that are come from mother, and some genes that have come from father. And when we say so, there will be, that means we have two copies of every gene. Say, for example, gene A. We have one from mother, one from father. And they may be expressed from both the genes. Okay, expression may happen from the both alleles. In some cases, expression may happen only from one of the alleles, either mother or father, randomly. And there are some genes where expression will only happen from only the maternal allele, or expression may happen only from the paternal allele. Okay, that means there is something about that particular allele that it is because of some modifications, which are epigenetic modifications, that will determine that the gene is either expressed or repressed, right? So let's see what genomic imprinting actually would mean. It's a epigenetic differentiation or modification. Usually, I hope you remember what is meant by epigenetic modification. It is any type of modification on the genome that has a consequence with the gene expression, but it should not be a change in the sequence. If there is a change in a sequence, base in the nucleotide sequence, then we would call that a mutation. But if there is any other mutation other than the change in the sequence, then we would call it would be epigenetic modification. Typically, the most common epigenetic modifications are DNA methylation and histone modifications. There are also others which we will determine, like the chromatin architecture, nuclear localization, and other things, which we will discuss in the later classes. And that is the first part of it. Genomic imprinting, the first part is it is a epigenetic modification. Second thing is it depends the expression. The modification is determined by the sex of the parent. If it is a male, some genes are imprinted. If it is a female, some other genes are imprinted. Okay, so let's see what is happening here. Here we have a mouse embryo, and okay, we will just discuss this as it is from here. Okay, the gametes are there, mature gametes. Assume this is the sperm and this is the egg, and then they fuse, and that is the process of fertilization, and zygote is formed. And here IC is imprint control region, say IC1 and IC2. In the sperm, IC1 is suppressed. In the egg, IC2 is suppressed. And in the IC2 of the sperm is fine. IC1 of the egg is fine. That means their expression will happen. So upon fertilization and then further development, they will grow into a blastocyst and eventually as a fetus or embryo. If you see the somatoplasm of the whole organism, right, the whole body, within all the cells of these bodies, we would find that IC2 is expressed from the male maternal allele. This is the maternal allele because it's coming from the sperm, right? This will be the paternal allele because it's coming from the egg. So if you see here, the IC2 is expressed from the paternal allele and IC1 is expressed from maternal allele. So if you see here, there will be one expression, that is one-time expression of IC2 and one-time expression of IC1, right? It's like kind of complementing. What will happen now is when the embryo forms its own germplasm, that is primordial germ cells, and at that time or around that time, all the imprints, the DNA methylation there in that primordial germ cells will be erased. And that is called as erasure. And when the primordial germ cells are forming the gametes, then new imprints are established. Say, for example, if this embryo is male and it produces sperm, and in that the IC1 of all the sperms will be methylated or imprinted. If this embryo is a female, then the IC2 will be methylated in all the eggs that it is producing. Okay, so there is erasure of imprints that happens during the formation of the germplasm or the primordial germ cells, and new imprints are established, the establishment of imprints, and that is determined by the sex of the embryo that we are talking about. If the embryo is male, there is one kind of imprinting. If the embryo is female, there will be another kind of imprinting, right? Of course, in the picture, the one that is giving rise to must have come from another embryo. Okay, just for illustration purpose, they have shown it together. We will also see multiple other figures, and hoping eventually to give rise to or hoping to understand it better. So if you understood half of genomic imprinting in this class, that is good enough. So we'll try to go by. The first one is, what is genomic imprinting? Genomic imprinting is an epigenetic modification, and the epigenetic modification is determined by the sex of the parent. That is, some alleles are expressed from maternal alleles, and some alleles are only expressed from paternal alleles. So here is a female mouse that is here, a female mouse, and the female mouse is having, say, for example, we'll call it gene A. Gene A, one is coming from, say, female, and the other allele is coming from the paternal allele, right? This is a biparental mouse. All mice are. So in some cases, there are two copies of gene A, but one of it is methylated, or we can call it as imprinted, and the other one is expressed allele. Rise to mRNA, then proteins, and then there will be some phenotype. Similarly, there is a male mouse. It also has the similar type of gene sets, pair of gene A, and one of the allele is methylated, whereas the other one is not imprinted, and therefore expression will happen. When does it become parent specific? So during gametogenesis, when this embryo was forming its own germplasm, the primordial germ cells time, when it is doing that, the imprints that these cells, right, in the primordial germ cells, would have been erased. All of it is, all the modifications that are present will be removed. Then new set of imprint would have been established. So what will happen is, if this is a male, we said this is a male, right? So in the male, we have what should be that this gene A, both the copies will be imprinted, methylated, for example. Whereas in the female, both the copies are not methylated, they remain as such. Okay, so why is there no methylation anywhere here? It's because this is a female mouse. Female mouse does not, in the female mouse, when it is forming the gametes, there is no imprinting made on this particular gene A. Why is there imprint that is on both these alleles? It is because this is a male mouse. The sex of the parent is this one, because it is making these gametes. That will determine that these genes should be methylated. Okay? So if you see, process whereby a gene or genomic domain exists in a state of epigenetic differentiation that depends upon its parent of origin. The sex of the parent will determine whether imprinting should happen in that particular gene or not. Okay, the same case. So now, see, if we think that these two are have mated, and then this is one gamete, an egg, and this is a sperm, they would have fertilized and given rise to an offspring. The same, assume there are two offspring. Now, if you look at the somatoplasm of this individual, we'll have one copy of unimprinted gene A and one copy of imprinted gene A. The same is the case with the other one. One copy from here, unimprinted, and one here, imprinted ones. Eventually, again, there will be one copy of 1x times expression of gene A in both offsprings. Right? I hope you understood a little bit about genomic imprinting. Eventually, we will see, why should there be anything like this? What is the purpose of this? And these kinds of questions will come, and we will try to address it, and we will try to sometime come that we might not know completely. You can always dig into literature that is there. You can also study about the question like, why should any concept like this be in these mammals or complex organisms? What is the use of it? How is it evolutionarily better to have such a system? Right? So different people have different origins, and it is still an open question, but this is a phenomenon that is there. And by studying uniparental embryonic stem cells or uniparental research has actually allowed the thorough understanding of these things. That's why it is an important, it is generally a concept. It is one of the major concepts in stem cell research, and therefore we have to read it thoroughly and try to have a basic understanding. As we will see in the course, many things are not completely understood. There will be some things that we still people are trying to work it out. We will learn to the general principles as much as we know, but we will leave some areas that we don't know yet, or there will be modifications into what we know already, as more and more research will provide answers. So if you see a pregnant lady, you can see that this is one set of imprinting generations, right? This lady has, in her somatoplasm, has imprints of her parents. And she has contributed to the formation of one baby. And that is the imprints made within her. She made those imprints, and it is in the baby, right? Along with another male. And this baby is a product of this mother and a male. And this baby is making new the germplasm, which is having the imprints made by the fetus. The body that we have are products of imprints made in our parents. The body of our children are made by us. The imprints they made in there are us. It's a good way of trying to see how complex biology is, because we think we are one body, but we are actually not one body. We are kind of continuum with our parents. They made the imprints, and we have those, and we make different imprints, and the children will get the next generation. Okay, why we discussed genomic imprinting as a problem is we have seen that the uniparental stem cells do not have, cannot give rise to complete organism. In androgenetic, we find that they are aggressively growing cancerous type, and they give to mesodermal lineages and so on. Or in the embryo stage, at least, the placenta grows much and the embryo is small, whereas in gynogenetic zygotes, they give rise to embryos wherein the placenta is undergrown while the embryo is overgrown. And also from the chimeric studies also, we can also find that AG cells are different, and GG cells or gynogenetic cells are different in their potential to give rise to. They should normally, if they are normal like any embryonic stem cells, should be able to give rise to any cell type. But then that is not what is happening. And the problem is because of genomic imprinting. So I'll try to write with this. I hope you will excuse me for the technical problem today. So if you see that, if you see there is a gene A and gene A, so I think it will not be, this is difficult to write, A and gene B, right? There is also another set where we'll say gene A and A, B, right? Another set A and B. If it were normal, one will be repressed, A will be repressed, and in another one, B will be repressed, right? So how much expression do we have? We have one, sorry, I'll write here. Oh, it's difficult. So if you see like this, this is, sorry, I will try to take this again in the next class, but today you try to understand. If it is a normal one, you will have one copy of A expression. One is repressed and one is expressed. And in the case of alleles B, one is repressed, one is expressed. So you'll have 1X expression from these normal zygotes. If it were uniparental, then you may have 2X A expression of 2X or 2X B expression. And that is not a good thing, right? Either there would be overexpression of some genes or underexpression of some genes. And in either case, it is bad for the embryo. So what I also want you to remember is that to use embryonic stem cells for therapy, there are problems. One of it is AG, and one of it is the genomic imprinting. That is the biggest problem, because this abnormal gene expression of the imprinted genes would cause severe problems in differentiation as well as cell cycle regulation. And if it is AG cells, they cannot be used because they are mostly they grow, they become cancerous, and they cause death to the donor body or embryo. So if we see embryonic stem cells as such, they are very useful for research. Majority of the genomic imprinting and the understanding of genomic imprinting has come from the uniparental stem cell research. They can derive into some certain cell types. Maybe they can also derive certain cell derivatives, like maybe platelets or some similar things, and which can then be used for therapy. The advantages is that they are technically efficient, they are ethical enough, at least, and most importantly, they are immunocompatible. And immunocompatible because if we take the gametes from the person to whom we want to give these cells, then because the donor is the same person, and therefore they are immunocompatible. And we also discussed about how uniparental stem cells are probably better than the SCNT. It is about the discussion based on the telomere size, right? The germplasm telomere size is bigger compared to that of the somatoplasm, at least in the adult stage. The disadvantage is imprinting problems, because of which gene expression issues and then improper growth and so on. The other one is either they become cancerous. Transformation here means cancer. If I didn't tell, I will repeat. There are transformation. The word is used in molecular biology or molecular genetics to represent the uptake of naked DNA. Yes, right? A bacterial cell takes up DNA, then it is transformation. And the other transformation used is a normal cell becoming a cancerous cell. That is also the phenomenon is referred to as transformation. So AG cells, androgenetic cells, usually become cancerous, whereas the GG cells or gynogenetic cells have poor growth. I would want you to read the chapter from there, and there are a few more things that we will discuss, but I would want you to start reading on that. There are some technical details. You will get through many things that are depicted here. Strontium chloride, cytochelacin B, mitomycin. There are practical things. I don't need you to read the whole chapter as such, but I want you to know what these mean. What is thyroid solution? What is it used for? Hyaluronidase, CZB medium, mitomycin C. You should know the molecular mechanism of these components and the context of why they are using. So there are also ways of how there are technical details given in the text about how to induce ovulation, make pregnant male serum gonadotropin, injecting, then using several of these, and then collecting embryos, oocytes, and then inactivation medium, and so on. So I want you to read those things and try to find out the uses in this particular context, and I want you to do it by yourselves to start with. The first objective of telling you to do it by yourselves is that you should become self-reliant. You're growing up, and knowledge is free. It's available in your hand with the mobile. All it takes is we know English, we know how to operate things and find out data or information. All it takes is our own motivation to go there and start doing it. If we want to do that, then we have to make you do that, and that is as an assignment. Before I even discuss any of this at all, I would want you to find it, and these are some of the questions that come up in CIA as 2-mark questions if there are. Okay. But this is a theory course, and we will read limited amount of techniques and so on, the principles mostly. Because we are not going to follow this course with a practical course, we are not going to really do all the read the how everything. We would rather read the principles of those things. That is what I want you to find out. So we learned about pronuclear transfer in this course. I would want you to also look at, we will see a video or so that shows how pronuclear transfer is done. But in the next class, we are going to discuss about analysis, characterization of AG and GG cells. As I already told you, I would want you to read, please note it down, read about PCR, read about primer designing. The basics, whatever we have discussed before, that should be sufficient. If you can do that, then you will have a easy pass through the next class. Okay. So what we will do now is we'll just quickly watch a video that I picked. It's a small video of how they are transferring into the pipette. Is the technician is taking the embryonic stem cells. You can see they are round morphology of embryonic stem cells also, right? And they are collecting these embryonic stem cells, several of them, and then the technician will bring some eggs into the frame. And here is the suction pipette, and they will hold the pipette. With the pipette, they will hold a blastocyst that is present. This is the blastocyst. By adding negative pressure here, they will hold the blastocyst. Now he will make a puncture into the blastocyst. And put in the cells in there. These are all very sophisticated techniques, and one would require a lot of experience doing these kinds of procedures. Okay, I'll stop here. And if you have any questions, I would like to answer.
Sir.
Yes, please.
The capacity, like say 16 to 18, they say, right? The number of cells in a particular stage of the zygote, that makes
Yes.
it a blastocyst or something else. So if we add more cells to it, will it hold the cells that we have injected, or wouldn't it cause some disintegration?
Okay, I think you meant in a blastocyst, how many more, what is the capacity, how many more can we do? Can we put in? Is that right?
Yeah, the travel goes like in powers of 2, right? So it goes exponentially, and that's how the cell also maturizes and divides.
The embryo,
We are
yes.
forcefully injecting a large number of cells inside, so wouldn't it
Yes.
cause disintegration?
And I think if I'm right, in a blastocyst stage, by the time they have reached nothing has changed there. It's only in the form of ICM. The number of cells that would form the epiblast, okay? Until then, in the ICM, the cells also multiply. So I think that, they would have taken a cell stage, blastocyst stage, where it is still about to grow and multiply more into ICM. In that case, if they have given, if you have added a few more cells of it, it doesn't change the count too much. Or at least what I can say is it's not going to matter so much, the numbers, whether it is multiples of 2 or not. All that matters is if the hypoblast or the epiblast has formed properly or not. The next stage is gastrulation, and upon that you will form the primordial germ layers. The numbers, yeah, I mean, at the same time, you cannot stuff the whole embryo, whole blastocyst with ICM, because I'm sure the blastocele also, the cavity that is present in blastocyst also may have some purposes. So I think they will only put in as many they can. They wouldn't overburden it. But they should put considerable amount so that the probability of the parts when the body grows and the embryo grows, there should be considerable amount of body parts, tissues made from the inserted ICM or the embryonic stem cells. Okay? But embryology
but
is
it
difficult.
is already packed
Yes.
with a lot of antrum, and there's high pressure inside the
No.
blastocyst.
It is not packed with. If you remember, there is blastocele. What is CEOL? It is a cavity that is present in the blastocyst. You actually, I hope you remember it. I'll try to find some pictures. I think in the last videos also we have seen a few where the blastocyst is usually has a lot of gap. I can't assume where, but you can see my screen, right? So this part of this is called as blastocele. It is actually much more bigger than this one. Okay, this is a very small portion of it. Usually is the ICM, and this is fluid filled place. You can say pressure and all, but when you make a puncture and you put in cells, there will be some liquid or fluid flowing out through the same hole. Okay?
Yes, makes sense.
Okay. But, well, this is something we will want experienced people to, or we have to watch more videos on what they do. But this is what is to the best of my understanding. Pressure is not going to build, especially because there is a hole, and if there is a pressure, that will be ejected out. Once you make a hole, then the pressure is normalized with the external, whatever is external will also be the same. Yes.
Yes, sir.
Any other questions? Okay, thank you. I will try to fix my